Hello, everybody, and welcome to Silence Abounds Science at Home, where we will discover home lab magic. In this channel, we will perform experiments demonstrating scientific concepts, principles, and reactions in chemistry, biology, microbiology, microscopy, and molecular biology. Welcome to my channel. Hello, and welcome back to another episode of Silence Abounds Science at Home, where we will discover home lab magic. In today's video, we're going to demonstrate the analytical technique of thin layer chromatography. And we're going to use this technique to separate and identify amino acids. Thin layer chromatography is similar to paper chromatography, and please check out my previous video where I introduced you to chromatography and demonstrated the most simplest form of chromatography, and that's paper chromatography, where I separated plant pigments. Thin layer chromatography is similar to paper chromatography, and it uses different polarity to separate components of a sample. The difference is that in thin layer chromatography, a thin layer of silica gel is used as the stationary phase, whereas in paper chromatography, we use paper. The silica gel is usually spread on a glass or plastic surface. For today's experiment, I've got silica gel plates. So I've got a thin layer of silica on a glass plate. The rest of the process is the same as paper. At the end, we visualize the separated compounds. We can use various dyes. We can use UV lighting. Certain compounds will fluoresce under UV lighting. In this lab, we're going to use ninhydrin. And we'll discuss about the ninhydrin reaction a little bit later when we come to spray the plates. So we spray the plates with ninhydrin in order to visualize the separated amino acids. Using the retention factor, we can compare our sample against the known standards to identify the amino acid in our unknown sample. Amino acids are the building blocks of proteins, and they play a crucial role in many biological processes. There are 20 standard amino acids that are encoded by the genetic code. They are based on their side chains or their R groups and can be categorized into essential, non-essential, and conditional amino acids. Essential amino acids cannot be synthesized by the body and must be obtained through diet. Non-essential can be made by the body, so they don't need to be consumed in the diet. Conditional amino acids are usually non-essential, but in certain situations like illness, severe stress, they may become essential. Amino acids have a central carbon atom, attached to an amino group, carboxyl group, a hydrogen atom, and have variable side chain or R groups. In this lab, we're going to separate and identify 3 amino acids: alanine, glycine, and leucine. And we're going to we have some known standards containing those 3 amino acids. And we have a sample that we know has an unknown amino acid. So we're going to discover or use thin-layer chromatography to identify the amino acid in our unknown sample by comparing it with our known control samples. The TLC plates, the thin layer silica gel plates, can be purchased anywhere. Any of my resources that I list in my description of my video, and also even on Amazon, you can buy both thin layer chromatography plates, and you can even get the chromatography solvent on Amazon. I have my chromatography solvent. I'm going to use isopropanol, but another common solvent that is used for chromatography is ethyl acetate. In addition, I have ninhydrin powder, which I'm going to reconstitute in alcohol just before I need to spray my plates. What I'm going to do is I'm going to have 3 separate plates, and each plate I'm going to spot one of the amino acid controls that we have, and then I'm going to spot the unknown sample on each of the 3 plates as well. I then am going to run them in, I got 3 separate beakers, which will contain my chromatography solvent, and then I will let them dry and stain. So let's get started. Okay, so before I apply the samples and controls to the chromatography plate, I want to allow my chromatography solvent to incubate and equilibrate in the beakers. So I'm going to pour in my solvent into each beaker. And it's important that you don't put too much that it goes over the solvent the application line. And I will explain that in a minute. So the application line is where we apply the sample. And if you put too much solvent into the beaker, into the chamber, what happens is the sample that you apply to the plate just dissolves in the solvent. So you want the solvent to be below your application line so that when it starts moving and migrating up the plate, it takes the sample with it, rather than if you put the plate in the solvent and the solvent goes over the application line, then in that case, the sample will just dissolve into the solvent, and that's not what you want. So just be careful and be mindful of that. Just so you don't really need a lot of solvent. And what is recommended in chromatography is just to equilibrate the chamber for a few seconds. No, it doesn't have to be a long time. So that will give us time to apply the sample onto the plates. And by the time we're done with that, the chambers will be ready for the chromatography. So I'm just going to put these to the side while they equilibrate. Okay. And then I have my samples here. So I have 3 standards, alanine, leucine, and glycine. And then I have my unknown sample here. So, so I'm going to apply now my samples and controls, and I'm just going to apply um, using these capillary sticks. I have tubes of capillary for each. So what I'm going to do is I'm going to apply the alanine standard, and you don't want to apply large amounts of sample. You want to keep the application area small, but you can spot it a couple of times. Okay, so I'm using a capillary stick to suck up my sample. You can use a pipette, a small micro pipette, and you just want to. Hold on a minute. You just like so. And that's bigger than I would like, but what you do is you just wait it to dry a little bit, and just you apply a couple of times. That's all you need. So I'm just going to apply a little bit more. Okay. Let that dry. Okay. So I'm going to apply like just 3 drops, 3 small drops to the application line. Okay, that's plenty. It's rather bigger than I would like. So if you can make the application circle a little bit smaller, that'd be great. So that's the first standard. So now I'm going to apply my other standards. And then lastly, I'm going to apply my sample to each plate. So I've now applied my controls and my unknown sample. So before I put them into the chromatography tanks for developing, you just want to make sure that each plate is dry, each application has dried. So you now just leave the plates to dry for a few minutes. And also, a very important point is when you're drawing, so what I have is my application line is 2 centimeters from the bottom. And then I drew a solvent front line, which is 1 centimeter from the top. And this is the line that my solvent will travel to. So once the solvent gets to my solvent level line, I take the plates out of the chamber. Okay. And it's important when you're drawing them to use pencil only. Do not use a pen or ink or marker, because the ink from the markers will migrate and travel in the sample. in the solvent. Okay, so you don't want that happening. So now I'm just going to leave my plates to dry a little bit, and then I'm going to put them into the chromatography tanks. So our samples have dried on the plates, so it's now time to put them into the chromatography chambers. Okay, so now it's important that you don't touch the plates or the area where the sample is or going to travel. And you just pop in the plate very gently, like so. Cover again. It's important you don't use your fingers or transfer any of your oil, skin oil, onto the plates, because that can hinder the migration of your compounds. So I'm going to put this in. So cover. And then for plate 3, again, okay. So we now then just leave the solvent to travel up the plate up to the solvent line. So you just have to keep an eye on it and then take out the plates once it's reached the solvent line. So the solvent has now reached the solvent line. So I've taken the plates out of the chromatography chambers, and now we have to let them thoroughly dry before we do the staining. Okay, so we've left the plates long enough and they should be completely dry now, and we can move on to the staining process. Today, we're going to stain our plates using ninhydrin reagent. Now, the ninhydrin reaction is a classic chemical test used to detect the presence of amino acids and proteins. The reaction involves ninhydrin, a chemical reagent that reacts with free amino groups. Ninhydrin is a powerful oxidizing agent and reacts with the amino group, resulting in a deep blue or purple color. This is commonly referred to as the Rubman's purple reaction. In some cases, a yellow color is observed, especially with proline due to their cyclic nature. But today, proline is not one of the amino acids we are looking at. So whenever you're handling ninhydrin, you must wear gloves. The reason being is because ninhydrin reagent will stain your skin because of the proteins in your skin and amino acids. And the stain will, it cannot be washed away, and it will remain on your skin for a long period of time until your skin starts shedding cells. So please, whenever handling ninhydrin, always wear gloves. Believe me, I've experienced staining my skin with ninhydrin, and believe me, you will be stained for a very long time. So I'm going to put my gloves on, and you want to dissolve ninhydrin powder in alcohol, in ethanol. So I have a spray container right here. And what I'm going to do is I'm going to add 4 bottles of alcohol into this container. And then it should mix and dissolve quite easily. And then what we're going to do is we're going to spray the plates with the reagent. Now I'm going to put the plates in a cardboard box and then spray it just so that the spray doesn't go everywhere, because you do not want ninhydrin spray going everywhere. So I've added the final bottle of alcohol, and then I'm going to give this time a few seconds of mixing to dissolve all the ninhydrin. Okay. Next, we need to set up the box to do the spraying. Okay, so I've got my plates lined up in this cardboard lid, which should be sufficient to prevent the ninhydrin going everywhere. And then what I want to do is do a small spray evenly across the plates. So I'm just going to squirt a little bit to prime. There we go. And then I just spray evenly across the plates. You don't want to saturate the plates, okay? And then you just leave that to develop. And leave enough time for the ninhydrin to react with the amino acids. Okay, so here are our results. This plate 1, for some reason, the alanine is not showing up. Maybe I didn't apply enough sample, but that's okay, because what we got here, this is our leucine sample, our leucine standard, and here is our sample, our unknown sample. And I got a spot, I can bring the camera closer. I got a spot here, and then I got another band here. You can't really see the distinction very well on the camera. And then on this plate, which is my glycine plate, again, I got 2 spots. It's a little bit more obvious here and then here. And then this is my standard. So looking at this, it looks like my unknown sample has 2 amino acids. This spot corresponds to the leucine standard, and this spot here corresponds to the glycine standard. So from this, just visualizing the results, you can determine that there are 2 amino acids present in the unknown. There's glycine and leucine. Now, if we hadn't run these standards, but we knew what the RF factor was, we can calculate the RF factors on the samples, on the unknowns, and compare it with known RFs that were previously calculated. Well, I hope you enjoyed that illustration of chromatography, thin layer chromatography for the separation of amino acids. Thin layer chromatography has a multitude of uses, and multiple components, compounds can be used to separate thin layer chromatography. There are other forms of chromatography, as I discussed in my previous chromatography video. There is also a column chromatography, which will be featured in a future video. There's also more complex, highly sophisticated forms of chromatography, including high performance liquid chromatography or HPLC, where components are separated through a column, through a liquid solvent. And then you have gas chromatography or GC, where components are separated in the column by gas mobile phase. Those are very sophisticated, very expensive, large instrumentations that can be found in clinical research pharmaceutical laboratories. I hope you enjoyed this video. Please hit the subscribe button if you're new to the video and you're viewing. Please hit the like and share button. Please subscribe because it helps me support my channel, making more videos, more experiments, more complex experiments. Please leave a comment. Let me know what you want to see. If there is a particular scientific principle or concept you want demonstrating, or a particular technique you want to see, please leave a message in the comments. Also, you can reach out to me by email, send me questions about anything about science, about my channel. Also, send me your videos and photos. I want to see you doing experiments in your own home. I want to truly build up a home lab community. Please check out my video description. I list all my resources that I use for my home lab that you can also check out and purchase items so that you can reproduce and do all the experiments that I do in your own home. With that, until next time, keep on experimenting. Goodbye, everybody.